When you take a living tissue and burn it, then only the inorganic salts and the elements are left. But here you are analyzing by dissolving in trichloroacetic acid. So all these elements, the salts will be found in the acid soluble fraction. Here, these are the inorganic constituent of the living tissues: sodium, potassium, calcium, magnesium, water, compounds. They are all inorganic, but they are found in the living tissue. You cannot classify them into biomolecules, but here they are the elements, they are the radicals, they are the salts which are found in the acid soluble fraction. Now, if you do elemental analysis, then you will find hydrogen, carbon, oxygen, nitrogen, chlorine. But we are doing about the biomolecules. If we do with the chemistry point of view, then you will find it as aldehyde, ketones, aromatic compounds. But with the biological point of view, you will find fatty acids, amino acids, nucleic acids. So here we are doing the analysis of biomolecules. So the molecules which are found in the acid soluble fractions, they are microbiomolecules. So they are carbon biomolecules, are carbon compounds. Here the structure which you can see is of glucose, C6H12O6. This is how it is arranged. This is the structure: CH2OH, OH, OH, and here the OH is on the other side and rest of the OH is downwards. This is how it is arranged in C6H12O6. Then this is the structure of the ribose sugar which you find in DNA and RNA. Here the structure is a bit different. HOOH and CH2 is arranged in this way, OH is downwards. So these are the sugars which are found in the carbohydrates. They are 180 Daltons. They are very small, so they are found in the acid soluble fraction. Now, as we conducted the experiment of dissolving the living tissue and TCA, we have seen the micromolecules which were there in the acid soluble fraction. Now, the one which are retained, which are insoluble, they are macromolecules. The one which is soluble in the acid, they are biomicromolecules. So we have studied biomicromolecules. Now let's see what is biomacromolecules. What is there in the retentate? The one which has the size more than 10,000 Dalton, they are in macromolecules. The one which is from 18 to 800 Daltons, they are in micromolecules. So here in biomolecule, that is acid insoluble organic compounds, are protein, nucleic acid, polysaccharide, lipids. Now protein, nucleic acid, polysaccharides, they are polymeric substance. They are polymeric substance. They have undergone polymerization. They are big molecule, more than 10,000. Now lipids have got very less molecular weight, but still it is up in the insoluble acid retentate. Now lipids are the only micromolecules which are not in acid soluble solution, but in acid insoluble substance it is held up. Lipids are the only micromolecule which behaves as macromolecule there in the acid insoluble retentate. Now the reason of it is Lipids are present in the cell membrane. When you have broken the cells, the cell membrane has broken. When it breaks, you know that cell membranes are made up like this, made up of phospholipids, which has got hydrophilic and hydrophobic structures which get attached like together. Now when you break it, they break open, but due to its tendency to come together, they arrange themselves to form vesicles. Vesicles, they become round in structure, they become vesicles, and therefore they are held up in the macromolecules. They are held up as macromolecules in the retentate. So this is the reason why lipids is there in the retentate. Acid insoluble substances. Now when you analyze this protein, nucleic acid, polysaccharides, all this substance, the quantity and the acid soluble substance and the acid insoluble substance together is the total number of organic compounds which are present in the living tissues. So you can see this chart. Water is the molecule which is present in the living organism to the largest extent. So here you can see the percentage of the substance in the molecular mass, cellular mass. Now water, it is 70 to 90%. Protein 10 to 15%. Carbohydrate 3%. Lipids 2%, nucleic acid 5 to 7%, and ions are 1%. So this is the comparative study of different molecules which are present in the living. Now students, you find protein in the insoluble acid insoluble fraction because they are polypeptide. They are linear chain of amino acids, long chain, polypeptide. It's a long chain, that is protein. And they are not homopolymer, they are heteropolymer. Homo means same unit and number of times. Hetero means they will be different unit and number of times. Now, there are 20 proteins in our body, amino acids in our body. Now some are essentials and some are non-essentials. Essentials are the one which we should have through our diet. Non-essentials we may get through the diet or our body will be able to synthesize. Some of the proteins among these 20 proteins: alanine, cysteine, proline, tryptophan, lysine. These are the proteins present in our body. Now proteins, they perform certain function in our body. Let's see what are those functions. Collagen. Collagen are the protein which are present in the intercellular spaces, intercellular ground substance. They give elasticity to the tissue. Trypsin. Trypsin is made up of 3 amino acids. They help in digestion of protein. They are proteolytic. They help in breaking the protein in the intestine. Insulin is a hormone which helps to maintain the glucose level in our body. Antibodies. They are the small cells which protects our body from the external microorganism. Those antibodies are also made up of proteins. They fight infection. Whatever infection is there in our body, antibody will fight. Supposing we lack protein, then our immunity is not good. We are not able to fight infection and therefore we remain sick. receptors, sensory receptors like smell, taste, all those receptors are made up of protein. Now GLUT4, that is glucose transport system 4th type, that enables glucose transport into the cells, is also made up of protein. Now other functions, proteins transports the nutrient across the cell membrane. Inter-exchange of the nutrient from the cell membrane is done by the protein. Now collagen. Collagen is intercellular ground substance, that is the protein which is maximum present, that is the animal tissue, present in the animal tissue, and that is the largest amount of protein in the animal tissue. Now the largest amount of protein which is present in this wall, you can say, that is ribulose biphosphate carboxylase oxygenase, that is Rubisco. Now polysaccharides, they are made up of monosaccharides. Monosaccharides, many sugar, 1, 2, 3, 4, many sugars, they will make polysaccharide. They are polymer, and they are homopolymer because monosaccharides are all same, therefore homopolymer polysaccharide. They are bigger in size. Since they are polymer, they are held up in acid insoluble fraction, and therefore they are biomacromolecules. Cellulose, the outer membrane of the plants that makes paper, the cotton fibers are all polymer, polysaccharide. They are held up in the acid insoluble fractions. They are made up of small sugars. Glucose are held up, that is cellulose. It's a sugar. Now starch is another sugar. It's helical in structure. If you put iodine, you will see blue color structure because helical structures, they have space in between to hold iodine. But if you put iodine in cellulose, it won't turn blue because there's no space in its helices. Now, glucose is present in the glycogen. The animals store glucose in the form of glycogen in the liver. This is the structure of glycogen. Now you might see it looks like a dry stick of plant, but here there are glucose, 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 glucose. The monosaccharides are held together to form polysaccharide. It has got, here is the non-reducing end, here it is the reducing end. You can see the structure of glucose here. It is in fact glucose, 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 glucose. Here you can see the structure of glucose. So this is the representation of glycogen. Now you find more complex proteins along with the amino group. So here the examples you can see, it is glucosamine and acetyl galactosamines. Now chitin, the covering of the arthropods, which is shiny in color, which gives protection, it is N-acetyl glucosamine. So all these complex polysaccharides are present in nature. Now inulin. Inulin is a fructose. It is fibers. It is probiotic. When you take wheat, you take banana, you take chicory, all this consist of inulin, which is probiotic, which is good for health because it helps in excretion and keeps our system healthy. So polysaccharides, they are long chains of sugar. They are chain of monosaccharides. Monosaccharides together they form polysaccharide. So cellulose is polymeric. That forms the polysaccharide of glucose. So starch is another form of glucose. They are all glucose, but the structure differs in starch, it differs in glycogen, it differs in cellulose. And here you can see the difference in the structure of starch, glucose and cellulose. Now, nucleic acid. Two types of nucleic acid, RNA and DNA. When it is made up of ribose sugar, it is RNA. When it is made up of deoxyribose sugar, it is DNA. Now, it is found in the acid insoluble fraction. Why? Because it's polymer, it is a big molecule, it is made up of nucleotide, many nucleotide, they will come together to form DNA. Now what this nucleotide consists of? You have already studied, it consists of a sugar. It consists of a ribose sugar, it consists of phosphoric acid, and it consists of a nitrogenous base, that is adenine. It might be adenine, thymine, guanine, cytosine, uracil. So here it consists of a nitrogenous base. It consists of a sugar, it consists of phosphoric acid, it makes a nucleotide. Now this is the structure of DNA. You can see many nucleotides. Adenine, sugar, phosphate. It is one nucleotide. Here again guanine, sugar, phosphate. Another nucleotide. Here thymine, sugar, phosphate. A nucleotide. Here cytosine, sugar, phosphate, nucleotide. So you can see one unit nucleotide, 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 nucleotide. Goes on, it forms a long chain. A chain of nucleotide which consists of a heterocyclic nitrogenous base, a sugar and a phosphate group. They form a nucleotide, and nucleotide is present in DNA, RNA. Now there is a hydrogen bond in between. Now this part, it forms a base pair. It forms a base pair. Here is the skeletal structure which is formed by the sugar phosphate. It forms a backbone. Here sugar phosphate backbone. Now adenine, guanine are purines. Rest thymine, cytosine, uracils are pyrimidines. So here nucleic acid, it is made up of nucleotide, and that makes it a big structure, and it does not comes into the acid soluble fraction, but it remains in the acid insoluble fraction. Therefore it is a macro. It's not micro, it's macro biomolecules. It's bigger in size. So nucleic acids are micro biomolecules.